Yeah.
Every now and then, you hear about a scientific leap, something that just feels, well, huge, maybe even Nobel Prize territory.
Uh-huh.
And right now, there's a lot of buzz around a new kind of drug. One that doesn't just, block proteins, it actually erases them.
Yeah, but what if the problem is simpler? Like just having too much of a protein, or maybe it's folded wrong, causing chaos.
So not just tweaking it, but getting rid of it entirely.
Precisely. It's really blossomed into this incredibly promising field. Targeted Protein Degradation, or TPD. And what's so fascinating, I think, is the fundamental shift in how we're thinking about disease treatment.
Let's dig into that. So these are molecules designed to basically point a finger at bad proteins and tell the cell, hey, recycle this.
That's a great way to put it. They're like molecular matchmakers.
Okay, so you're hijacking the cell's own cleanup crew.
Exactly. It's quite clever.
And this is where it gets really exciting for diseases like Alzheimer's, right? Because we're not just talking about, say, managing symptoms anymore.
That's the potential game changer. For ages, the buildup of proteins like amyloid beta and tau in the brain has been the prime suspect in Alzheimer's.
We've heard about those for years.
We have. And finding ways to clear them out safely without damaging healthy stuff,
Yeah.
that's been the massive hurdle.
Right. So our mission today, unpack what targeted protein degradation really is, explore the different ways it works inside our cells, and understand why everyone's saying it could be such a game changer for treating a whole range of diseases, including those tough ones in the central nervous system.
Okay, let's dive in. You mentioned traditional drugs usually block protein activity. So what's the absolute core difference with TPD?
Well, like we hinted at, it really boils down to the end goal. Instead of just putting up a roadblock, TPD aims for complete removal.
Get rid of it entirely.
Yeah, think of it less like turning off a light switch and more like actually taking the bulb right out of the socket. And the clever part is it does this by kind of hijacking the cell's own built-in garbage disposal systems.
It's amazing, really. We're basically learning to direct the cell's own quality control.
What exactly are PROTACs?
Okay, so PROTACs are a key part of this newer field called Targeted Protein Degradation, or TPD. The core idea is they're what's called heterobifunctional molecules.
Heterobifunctional. Okay.
It sounds complex, but think of it like a molecule with two distinct hands or binding domains.
Two hands. What do they do?
So one hand is designed to grab onto the target protein. That's the protein we want to eliminate. Maybe it's misfolded or aggregated, causing problems.
Like the tau protein in Alzheimer's.
Exactly like tau or alpha-synuclein in Parkinson's. The other hand is designed to grab onto something called an E3 ubiquitin ligase.
E3 ubiquitin ligase. Right, that sounds important, but technical, what's its role?
It's a key part of the cell's natural protein disposal system. Think of it as the cell's tagging machinery, part of the ubiquitin-proteasome system, or UPS.
Tagging machinery, like putting a label on something.
Exactly. Its job is to put a specific molecular tag ubiquitin onto proteins that are damaged or no longer needed, marking them for destruction. It's like the cell sticking a dispose of this note on the protein.
Okay, so one hand of the PROTAC grabs the bad protein, the other hand grabs this tagging E3 ligase enzyme.
Yes. The PROTAC acts as a bridge or a matchmaker, forcing these two proteins, the target and the E3 ligase, to come together when they normally wouldn't interact much. That's the chimeric part.
It brings them together, and then what happens?
This forced proximity is the key. It tricks the E3 ligase into putting that ubiquitin tag onto the target protein.
Ah, so the PROTAC doesn't do the tagging itself, it just makes the E3 ligase do it to the target.
Precisely. The PROTAC induces the tagging. Think of ubiquitin as that kiss of death signal for the protein.
Okay, kiss of death. Protein gets tagged. What's next in this cellular disposal line?
The ubiquitin tag is then recognized by another massive piece of cellular machinery called the proteasome.
The proteasome.
You can think of the proteasome as the cell's molecular shredder or recycling center. It finds proteins tagged with ubiquitin and it just completely degrades them, breaking them down into small peptides.
Wow, so the PROTAC is like a highly specific guide, leading the cell's own shredder to the exact protein we want gone.
You've got it, and here's a really critical point. PROTACs are catalytic.
Catalytic, meaning?
Meaning one PROTAC molecule isn't used up in the process. After causing one target protein molecule to be tagged and degraded, it can detach and go find another target protein molecule, bring in another E3 ligase, and repeat the process.
Whoa, so a single PROTAC molecule can take out many, many target proteins.
Potentially, yes. This allows them to be effective at very low doses, what we call substoichiometric levels. It's incredibly efficient.
That sounds fundamentally different from most drugs that need to bind one-to-one with their target.
It is. It leads to what's called event-driven pharmacology,
Event-driven,
as opposed to traditional occupancy-driven pharmacology.
Hmm.
Most drugs, like inhibitors, need to constantly occupy their target's binding site to work. If the drug concentration drops, the effect wears off quickly.
Right.
PROTACs trigger an event, the destruction of the protein. Once the protein is gone, it's gone, at least until the cell makes more. So the effect can last much longer, potentially, even after the PROTAC molecule itself has been cleared from the system.
That pharmacokinetic-pharmacodynamic disconnect you mentioned. The drug leaves, but the effect remains because the target is physically removed.
Exactly. That could mean less frequent dosing, maybe. It's a very different profile.
And you mentioned something about a ternary complex.
Ah, yes, the stability of that three-part complex, the PROTAC, holding on to both the E3 ligase and the target protein simultaneously is crucial. If that handshake isn't stable enough, the ubiquitin tagging doesn't happen efficiently. So designing PROTACs that form stable ternary complexes is key.
Makes sense. Like a good, firm handshake is needed to pass the message along.
You could put it that way, and maybe one of the most exciting parts, especially for neurodegenerative diseases, is that PROTACs don't necessarily need a traditional active site to bind to.
Ooh.
Think about those protein aggregates like amyloid plaques or tau tangles. They're often just messy clumps. Traditional inhibitors need a well-defined pocket to fit into, which these aggregates often lack.
Right, they're just blobs.
Pretty much. But a PROTAC just needs somewhere on that protein or aggregate to bind, even a relatively featureless surface, to bring the E3 ligase nearby. This opens the door to targeting things previously considered completely undruggable.
That's huge. So targeting those notorious aggregates in Alzheimer's or Parkinson's suddenly seems more feasible.
That's exactly the hope and the focus of a lot of current research.
Okay, this really sounds revolutionary. How do PROTACs compare then to the therapies we're more familiar with, like standard inhibitors or maybe gene therapies?
Good question. Let's start with standard inhibitors. As we said, they block a protein's activity, usually at one specific site.
Like putting gum in the works.
Kind of. But the protein itself is still there. It might have other functions or just be taking up space, maybe interacting with other things in detrimental ways, even if its main activity is blocked.
But PROTACs.
PROTACs destroy the entire protein scaffold, so all its functions, all its interactions are gone, kaput. And again, they can hit targets that inhibitors simply can't bind to effectively.
Total removal versus just silencing one function. Clear difference. What about gene therapies? They aim to fix the problem at the source, right? At the DNA level.
Exactly. Gene therapies try to prevent a faulty protein from being made in the first place, or maybe introduce a correct copy of a gene.
So stopping the factory production line?
Right. PROTACs, on the other hand, work on the proteins that already exist in the cell. This is important because sometimes the problem isn't just the protein itself, but how it gets modified after it's made these post-translational modifications.
Like phosphorylation of tau.
Precisely. PROTACs could potentially be designed to selectively target those disease-modified forms of a protein, leaving the normal ones alone. Gene therapy doesn't really distinguish once the protein is out there.
I see. So gene therapy is prevention at the blueprint level. PROTACs are cleanup of the existing, potentially faulty products. How about antibody therapies? They're big players now.
Antibodies are fantastic tools, but they are large proteins themselves. They generally work outside the cell or on the cell surface. Think immunotherapy targeting cancer cells or antibodies trying to clear amyloid plaques outside of neurons.
Okay, so mainly extracellular targets.
Mostly, yes.
Yeah.
PROTACs are typically smaller molecules designed to get inside the cell. And for many neurodegenerative diseases, the real culprits, the tau tangles, the alpha-synuclein aggregates, are accumulating inside neurons.
Where antibodies struggle to reach effectively.
Exactly. So PROTACs offer a way to directly target these intracellular pathogenic proteins, which is a huge advantage for CNS disorders.
Can you give some specific examples from the research in neurodegeneration?
Definitely. Let's talk tauopathies, diseases like Alzheimer's and frontotemporal dementia, FTD, where abnormal tau protein is the main problem. It misfolds, clumps together, forms those neurofibrillary tangles, and becomes toxic to neurons.
The hallmark pathology we always hear about.
Right. And studies have shown that PROTACs can be designed to get into cells and specifically degrade this intracellular tau. Different research groups have shown this works using various E3 ligases, VHL, CRBN, even KEAP1.
So multiple ways to tag tau for destruction.
It seems so. And there's a specific example of PROTAC called QC01175. In lab models using neurons derived from FTD patients, it showed preferential degradation of the aberrant disease-associated tau over the normal, healthy tau.
Wow, selectivity. That's the dream, isn't it? Getting rid of the bad stuff while leaving the good stuff alone.
It's absolutely critical, especially for proteins like tau that also have important normal functions. And the work didn't stop there. Follow-up studies have focused on optimizing the linker part of the PROTAC, the bit connecting the two hands.
Fine-tuning the bridge molecule.
Exactly. They've worked on improving the degradation of the really tough, insoluble tau aggregates found in tangles. It shows the field is actively working on making these molecules more potent against the most stubborn forms of the protein.
That sounds incredibly promising for Alzheimer's and related dementias. What about Parkinson's disease? That involves a different protein, alpha-synuclein.
Right. Parkinson's and related synucleinopathies involve the abnormal accumulation and aggregation of alpha-synuclein, forming Lewy bodies and Lewy neurites inside neurons.
Another intracellular menace.
Indeed.
Okay, this all sounds incredibly exciting, but it can't be easy. What are the major hurdles, the key challenges in actually getting PRO to work effectively as drugs for brain diseases?
You're right, the potential is huge, but so are the challenges. Molecular design is probably the biggest one.
How so?
Well, PROTACs, being these two-headed molecules connected by a linker, tend to be quite large compared to traditional small molecule drugs.
And larger molecules generally have a harder time crossing the blood-brain barrier, right?
Exactly. These larger molecules have unique challenges. They need to maintain enough solubility, engage both targets effectively, and have the right properties to sneak across the BBB. It's a very tricky balancing act.
You mentioned VHL and CRBN are commonly used.
They are, almost exclusively in clinical development so far, but there are hundreds of E3 ligases in human cells.
Yeah.
Relying on just two ubiquitously expressed ones like VHL and CRBN carries risks.
Risks like
Potential toxicity. If you're hijacking a widely expressed E3 ligase and your PROTAC isn't perfectly specific for the target protein, you might accidentally degrade other essential proteins, off-targets, that also interact weakly with that E3 ligase via the PROTAC.
unintended consequences.
Exactly. The big hope is to find and develop ligands for E3 ligases that are more tissue-specific, perhaps enriched in the brain or even in specific neuron types relevant to a disease.
That would give you another layer of selectivity. Are there known brain-enriched E3s?
Yes, research is identifying them. For example, RNF182 seems to be upregulated in Alzheimer's disease brains. There are Kelch family E3s like ENC1 expressed in the brain. Also, some neuronally enriched TRIM family ligases like TRIM9 and TRIM67 are being explored. If we could hijack those specific ligases, the PROTAC would theoretically only work in the cells where that ligase is present.
That would be much safer. So molecular design for BBB and expanding the E3 toolbox are key. Anything else?
Well, the BBB penetration remains the overarching challenge for CNS delivery. And related to the E3 ligase issue is the general concern about off-target protein degradation, ensuring the PROTAC only removes the protein you intended to remove.
Right, specificity is paramount. Okay, let's try to summarize this. The key takeaway seems to be that targeted protein degradation represents a truly significant shift in drug discovery.
Absolutely. It's offering a way to go after proteins that were simply off-limits before, which is incredibly relevant for complex conditions like neurodegenerative diseases.
And the potential impact, particularly for patients suffering from diseases like Alzheimer's, Parkinson's, Huntington's, it feels immense. This isn't just symptom management, it's potentially addressing the root molecular causes.
That's the ultimate goal and the promise that drives all this research. The idea of selectively removing toxic proteins could genuinely transform patient lives.
Just imagine if we could reliably develop therapies that selectively seek out and destroy the specific toxic proteins, the tau tangles, the alpha-synuclein clumps that are driving diseases like Alzheimer's and Parkinson's. What new possibilities for not just treatment, but maybe even prevention could that unlock? And what's the next crucial piece of research needed to take us closer to that reality? Something to think about.